I'm going to hide everybody's faces for the moment. Okay. Um, so this is an exciting collaboration between my lab and Ming's lab, and I'm just going to emphasize a few points. One is that Ming is the real developmental biologist whose aim is to define fundamental mechanisms of tissue patterning in development and regeneration of the skin. And the lab uses many different animal models. And as you'll hear shortly, we chose for the current project to use chick skin. Um, I think, I suspect the field is really uh, is just glimpsing this an enormously complex signaling network, which is operating across many scales in terms of both space and time. It's also involving quite a range of types of signaling, whether they be biophysical or biochemical. My role in this right now in this collaboration with Ming's lab is to look at bioelectricity in development. And we think that the ion channels play roles in the formation of competent morphogenetic fields. That in turn enables periodic patterning of appendages uh, within the fields. And furthermore, after the patterning has been laid out and the appendage primordia appear, the appendages are shaped by the function of these ion channels. And my curiosity leads me to wonder, how does bioelectric signaling fit into this very complicated signaling network that orchestrates the intricate development programs? And in particular, how does crosstalk occur between the other types of signaling? And I mentioned that we'd be working on chick skin. There are several very useful reasons to work on chick. One is that, can you see my cursor?
Yes.
Okay. One is that the feather buds, which we are going to study, appear in very distinct regions on the chick. So this is the dorsum, the back of the chick, and you can see indicated by these kind of dots patterns here. These are specific distinct tracks. This is exactly where the buds develop. There are also regions where there are no buds, and these are called apteric regions. And in the right, in the photograph, you can actually see, looks like periodic patterned buds. And in addition, you see a region where there are no buds, the so-called apteric region. This allows us to compare skin, adjacent to skin, with either with the buds or no buds. And as I'll mention later, we also have the exciting observation made by Hans that we can cause buds to form even in the apteric regions, and we're starting to investigate what's going on there. The other reason to work on chicken skin is that it's readily accessible to experimentation and explant culture. Furthermore, it has clinical relevance to wound healing and regeneration. So, well before I came into this collaboration, Ming's lab had already become very interested in skin bioelectricity for several unexpected findings in their experiments. One was that applying a voltage pulse in order to do electroporation, they observed that feather bud orientation could be disturbed, and I'll have a slide on this in a second. They also noticed that there is very stage specific and region specific expression of ion channels using bulk and single cell RNA-seq. Furthermore, they also had found by doing injection of dyes that travel through gap junctions that there is extensive gap junction coupling among cells in the epithelium and also among cells in the mesenchyme. This kind of coupling creates what is known as a functional syncytium. So the cells are electrically connected to each other. They still maintain a separate identity because they each have their own nucleus, but they are functioning as a unit. And I'm going to be talking a lot about this. Um, we also know from that early work that blocking the gap junctions changes pattern formation. This is just a slide to show how applying an external electric field can change the pattern in the back. You can see in the dorsal region of chick skin in an embryo, and you can see instead of having all of the feathers lined up as they normally are, pointing down the back of the chick, here you can see a whorl pattern. And each of the feather buds can be depicted as shown below on the left by an arrow, which shows both the direction of the feather growth as well as the length. And on the right is just illustrating that indeed, depending on the direction of the applied field, you can get different effects on feather bud alignment. So this is where this figure shows the observation that led Ming to invite me to start to work on this project. On the left, again, you see a chick embryo and a photograph of chick buds. The left is anterior and the right is posterior in this. Each bud is about 200 microns in the short axis. And what they had done is asked a collaborator, Min Zhao, to measure the electrical currents in the extracellular solution adjacent to the tissue. They were using what's called vibrating probe. I'll be talking about this a little bit more in the subsequent slides, but for this slide, the key thing to know is that the vibrating probe allows measurement of currents close to the surface of the skin. As depicted in the lower right, the arrows here of different colors point either in the upper panel downwards into the skin, or if you look at the next panel, you can see a blue arrow, which is coming away from the skin. So these are two different stages. These are showing that we get stage-dependent differences in whether the currents are flowing into the skin or out of the skin. All right. And blue represents the currents at the anterior, and green represents currents that are in the posterior of the feather bud. And in this case, as I'll emphasize, we've chosen to illustrate the currents at two really distinctive stages, H&H 31 and H&H 35. And in the graph to the left, what is depicted is how the current direction and amplitude changes as a function of time. I need to explain that in the electrophysiology convention, positive current, so if we look here, we have current amplitude in microamps per centimeter. We have the H and H stages on the top. 0 means the current, there's no current, obviously, and that's depicted with this pink line here. Currents that are above this 0 line are currents that are flowing out of the skin. Currents that are below this pink line are currents that are flowing into the skin. And what you see, for example, at stage 31 is the currents, whether we're looking at the anterior, and that's blue here, or posterior, that's green here, the currents are both flowing in both locations into the skin. But notice at stage 35, we have a separation. The blue, the anterior, has currents flowing out of the skin, and the posterior has currents flowing into, and it turns out this, in this case, to be the feather bud tip. I want to highlight the fact that these are stage specific and they're highly reproducible from tissue sample to tissue, a new tissue sample. There is some slight variation. That's because the staging is very approximate, but the important thing to note is that around stage 31, the currents are inward. At stage 35, they're outward. And if you look a little bit later, stage 37, 38, the currents flip. So what was initially an outward current or an inward current has flipped direction when the anterior and posterior. I'm going to come back to this. We were puzzling over this for quite a while, and now we have hypotheses about what's going on. I'll come back to this after I give a little bit of an introduction to what vibrating probe is and another technique that we are using called macro patch loose seal recording. I'm spending a few minutes on this because these are key to the experiments that we are doing. When we have electrical current flowing through a resistive medium, like physiological saline, a voltage drop will occur along the path of the current. That's simply Ohm's law. If we then use a voltage meter and measure the voltage at two separate locations along the direction of current travel, we'll get a voltage difference. As I said, this is really Ohm's law. But we can measure that current amplitude, and because the two electrodes are lined up either along the direction of current travel, or depending on how we angle it, we can get different fractions of the vectorial current. We actually measure the current amplitude, and we can actually get the direction of the current. Now, in the types of experiments that we're doing, we don't actually use separate electrodes here. It turns out that each electrode has problems with that we call them bias currents and difference currents. And it's better, in fact, to use one electrode and move it to different locations. So, for example, instead of having two separate electrodes, we could measure a voltage here and then another voltage here and calculate the difference. And again, use Ohm's law to calculate the current. In fact, what is done in the vibrating probe technique is that electrode, the same electrode, is moved side to side. We built one of these in the lab after Min Zhao's type of system. This would give us similar information to what would happen if we had two separate electrodes. Okay. Now, subsequent to this, we bought a commercial system. It's called an SVET. It's scanning vibrating electrode technique system. In this case, the vibration of the electrode occurs in 3 dimensions, and that information yields again vectorial information about the direction of current in 3 different axes, X, Y, and Z. This is what the overall system looks like. You don't need to read the labels. I'm going to zoom in on the working part of this system. So this is really the working part of the system. We have a stage where the explant, in this case, we're taking a piece of skin from the chick and we're attaching it to a working surface. There is an electrode, which is called here the probe, which is positioned near the skin surface. That probe is vibrated, as I said, in 3 dimensions by X, Y, and Z piezos. Piezos are crystals that deform according to the voltage applied to them. They can do this very quickly in the nanosecond timescale and reproducibly. We have 2 cameras that we use to visualize what we're doing. We have a top view to give XY information of the location of the probe relative to the skin. And then we have a side view to make sure that we don't crash the probe into the skin. So it enables us to put the probe close to, but not touching or penetrating the skin. Anyway, this is the general layout of our system. And in our subsequent experiments, you'll appreciate that this gives us directional information about the currents. The other system we have is the so-called macro patch setup. This is really based on patch clamp, and on the upper of these 2 diagrams here, I have a simple box called patch clamp amplifier. This is the box that applies a voltage to an electrode and then records the current that is flowing from a cell. So this round thing here is the cell. The pipette is here, and in this case, the pipette has been attached to the cell and attached mechanically and electrically tightly. and the membrane has been broken so that it's possible to record currents flowing through the cell. The output is a voltage which is proportional to the size of the current. Now, in what we're doing, it's called macro patch loose seal system. We are not applying the pipette tight to get this really tight seal to the cell membrane. We're simply using a pipette which has a much larger diameter than what is used in patch clamp recording. In this case, the tip of this is on the order of 50 microns to 100 microns. We're applying this a short distance above the chick skin so that we can record currents flowing near the skin. We lose some of the directional information about the 3D information, but this approach is very quick to set up. We can scan the surface and look for inhomogeneities in the direction of the current. Again, we have to look with two cameras to ensure that we maintain uniform distance above the skin. And this is a photograph of Hans and Dro working on a recording with the macropatch set up, and you can actually see here the image of the chick skin that they're recording from an oscilloscope to do the display during the live recording. And over here is the microscope, and the chick skin is there, and you can actually see the patch clamp head stage here and the electrode. So I left the other slide with this observation that the currents at the skin surface change at specific stages. And so this is what I was presented with when Ming came to talk to me. And now is about 10 years ago. I think we were at a dinner hosted by the MD PhD program, and we were just chatting. And he said, I have these puzzling observations. We don't really know what to make of them. So he showed me these drawings based on Min Zhao's data. And I looked at that and immediately said, well, the green and blue represent the currents flowing at the posterior or anterior of the bud. But, the skin. And to me, coming from, engineering and biology, biophysics background, it suggests that the current is probably flowing in this kind of a loop. So this is what I call a standing current. It turns out that, this return current here, number 4, can actually go in many directions. But for analyzing this, we can think of this as one small current loop. We've broken it into 4 different segments. And again, coming from a classical electrophysiology background, we have an appreciation for what type of channels can drive currents in these different places. The current that flows here in this segment 1 through and into the tissue is most likely, based again on work from Hodgkin and Huxley and others, is most likely a calcium or sodium channel. Current flowing through tissue, it either flows around the cells or it's going through gap junctions that are coupling cells. Ming's lab's early work already suggested that the cells here are coupled with gap junctions. Okay, and then in this point here, the segment 3, the current is going from the tissue and out into the extracellular fluid. Again, from classic work, I would have guessed immediately that this is either potassium or chloride channels. Most likely, I would have guessed potassium. And then the current, as I said, flows through the extracellular fluid to complete this loop. Okay, so this was the hypothesis, and you'll see we are excited because it explains a number of things. One is this kind of loop facilitates long range, even tissue scale signaling when the cells are coupled in with gap junctions in a functional syncytium. This kind of standing current can inject calcium deep into the gap junction coupled mesenchyme. It does require that there are different channel types, some located at widely separated locations they work together as a unit to sustain the current, the standing current. And I'm just going to go through some of the evidence for the standing current. Yes, we do have channels that are located, expressed at stage specific times. They are based on in situ hybridization and immunocytochemistry. They are located pretty much where the classical electrophysiology predicts. Furthermore, we can image calcium waves that originate from near the feather bud tip and propagate deep into the feather buds. We can do patch clamp electrophysiology of dispersed mesenchymal or epidermis, and we can record the currents. And indeed, they have calcium currents. And we use vibrating probe to measure the currents, and then we apply channel block to look at the effects on this standing current. I'm focusing on calcium for a very important reason. Calcium is a divalent cation, but it acts not only as a charge carrier, so it carries ionic current, but it's also a major second messenger. In this setting of development of the feather buds and probably more generally other types of development, calcium probably plays an important role by triggering secretion of, in this case, we're really keen on looking at and understanding how morphogens might work. And calcium is also found to play an important role in triggering cell motility, working through atypical myosins. And in the case of our functional syncytium, we can get whole collections of cells moving together, okay? And then calcium can also act on a range of calcium-dependent proteins and sometimes enzymes, for example, PKC, calmodulin, or calcium-activated potassium channels. Okay, so calcium is really key to playing a lot of important roles that are relevant in development. Calcium can also modify gene expression, okay? There are other roles of calcium that I'm not going to really talk about, but for example, calcium also triggers skeletal and smooth muscle contraction. And what's cool about calcium as a second messenger is that there are a lot of means whereby it can crosstalk with the cyclic AMP system. So here's some evidence that ion channels do play a role in featherbud. These heat diagrams show at two different stages. So the upper half here is stage H&H 31, and the bottom half here is stage 35. And each of these in vertical columns represents expression of different channel genes. And red is high expression, blue is low expression. And you can appreciate that there is a big difference in expression levels going from stage 31 to 35 of many of these channels. This is epithelium, and here is mesenchyme. But both epithelium and mesenchyme show very stage-specific changes in the types of channels being expressed. And using in situ hybridization, and later I'll show you some immunocytochemistry, we can start to dissect and figure out locations of some of these channels. So one of the voltage-gated channels is this is one of the voltage-gated channels here, shown, by the way, at different stages, 31, 34, and 35. You can see that it's expressed very highly in the epidermis, but also quite highly within the mesenchyme. Then we have the gap junctions, which are again expressed both in the epithelium and in the mesenchyme. Then we have stim and ori. Stim and ori together form what's called I crack. So that's a current that is, it's calcium release activated current. That's what the crack stands for. These are channels that feed calcium through the plasma membrane to refill calcium stores once the stores have been emptied. And then here we see evidence of a potassium, specific potassium channel in the anterior of the feather bud. So these early experiments done by Ang Lee really supported the general ideas that classical electrophysiology predicted. So the channels are kind of where we think they should be in order to sustain this standing current. Calcium is really indeed a big player. And the question is, what's the evidence that we actually have calcium going through? And now I'm going to dissect these segments 1, 2, and 3. But what's the evidence that indeed that there's calcium entering here? Well, we have the same series of experiments here. And this is one of Ang Lee's experiments and Zhonghua from my lab. They took the chick explant, the skin explant, and they cut a strip so that they could image from the side. Okay, and these are, again, different stages. There's stage H&H 31, 34, and 35. And they're measuring calcium by first transducing with the RCAS virus a combination of two reporters. One is called GCAMP, which is a calcium-sensitive fluorescent protein, which is depicted as green here. It is, in fact, a green fluorescent protein. And M-cherry, which is a red protein. The GCAMP reports changes in calcium, and that is normalized to the M-cherry level, so essentially to normalize to protein expression, which can be variable from cell to cell. So a ratio of these two plotted together gives some indication of how high the calcium level has risen, okay? And in this image, you can see the dotted line actually is the so-called range of interest, but this is in the mesenchyme, and above it is the epidermis, okay? And you can see this is a figure taken at the peak here. So what you actually observe is that if you stimulate with potassium chloride, extracellular potassium chloride, the cells are expected to depolarize. If they contain voltage-gated calcium channels, then we should see a rise in intracellular calcium as calcium goes through voltage-gated calcium channels. And what's interesting in the image of these cells while they're being stimulated is that the green is not present equally everywhere. Instead, we see regional elevations. So the anterior is to the left and the posterior is to the right. So we can see that the localized calcium elevation is mostly in the posterior distal bud. Okay, and this is really curious. It suggests that the cells within the mesenchyme may be heterogeneous. Yeah, they're all called mesenchyme or dermis, but the cells have different functions in different locations within this functional syncytium. One other experiment I want to highlight is this is some imaging evidence for eye crack. So that's that calcium release activated channel. And in this experiment, the ratio of the green to red fluorescence is plotted versus time. And the cells are exposed first to a zero calcium medium anthapsigarg, and that serves to empty out intracellular calcium stores. Then the external medium is replaced with a high calcium medium, or actually normal calcium. And what happens is with the delay, there is a sudden rise in calcium. And this is what's exactly expected for calcium that is flowing through eye crack. And we can block this with appropriate blockers of eye crack like BTP2, and it will essentially block this rise here. So we have evidence for both voltage-gated calcium channels and eye crack from these imaging experiments. We can also disperse the mesenchymal cells and patch clamp them. So this was done by Zhonghua from my lab, and this is a pretty it's a decent recording of voltage-gated calcium channel currents, and this is the so-called IV curve. I won't go into much detail except to say that not every cell had voltage gated calcium channels. About 30% of them had this voltage gated calcium channel. About 30% of them had iCrack with no voltage gated calcium channel. And then about 30% had both. And then there were about another group of cells, some of which had no voltage gated or iCrack calcium current. So there are at least 4 distinct populations among the mesenchymal cells. So I'm kind of excited. I've worked on calcium my whole career, calcium triggering exocytosis. But why is it a big deal that the calcium is entering through the tip of the feather bud and then propagating? And what's going on, I think, is that calcium serving these different roles that I kind of hinted at earlier. One is it facilitates collective cell motility. So here we have the feather bud. The lower part, which is more dense in cells, that is the mesenchyme, and on top is the epidermis. What you can see is that this is imaging over the time course of many, many hours. But the cells that are being subjected to this calcium, repeated calcium waves, they are actually moving. And there are 2 ways you can see this. One is that where the nuclei were originally located is where these green circles are. And these arrows, which change color with position, indicate the direction of movement. Okay. And you'll see at the bottom, there's some reddish. Actually, there's some blue right at the very bottom, but you see these colors become progressively red and then yellow. So that's a function of time. So this indicates that these cells are moving with time. Okay. And we can block a large part of this movement by applying blockers to the atypical myosins or blocking calcium entry. The other reason why the calcium entering the tip is a big deal is that, as you can see here in the in situs, the sonic hedgehog expression is confined pretty much to the epidermis right at the tip of the feather bud. Okay. And the receptors are located deep to that, but also in the epidermis. Now, this is kind of a cool observation because, of course, sonic hedgehog plays a huge role in triggering Wnt pathway and ultimately gap junction expression. So it's really cool that the calcium entry leads pretty quickly to arriving at the places where sonic hedgehog is produced and stored. And sonic hedgehog in different model systems is secreted on calcium elevation of the cytoplasm. Another reason why calcium entry near the tip is important is that there are so-called calcium activated potassium channels. It's hard to see here, but this is a feather bud at different stages. And what you see is, again, left is anterior, right is posterior. And this, you see stage specific expression of potassium, calcium activated potassium channels. They appear and then disappear, but they're very close to the posterior of the bud. So these are 3 processes that are facilitated by calcium entering from the feather bud tip and propagating towards the base. So I mentioned that there are gap junctions coupling the cells in the mesenchyme, and they're really important because they help this tissue to become a functional unit. They enable the current to pass along the length of the gap junction coupled tissue. And as I'm about to show you, not only do they pass electrical current, but it's possible that they also transmit an inhibitory signal for feather bud formation. So I can't go into, this was a long and detailed paper. I just show you that when you block the feather buds with a blocker of gap junctions, that there are supernumerary. So these buds start appearing in abnormal locations, and they don't appear everywhere, but they appear in certain characteristic locations. The the idea that the lab suggests is that there is some kind of an inhibitory um, agent that is transmitted through gap junctions, and if you block it, then new buds can form uh, in specific locations. Okay, so the previous part of this talk was old data. Now I'm going to show you data that is relatively new. We started to ask the question, okay, what evidence do we have that there are potassium channels here, and what happens if we block the potassium channels? What would the potassium channels in this location do? Well, they enable exit of the standing current from this mesenchymal cell collective to pass out of the feather bud. The other thing that they do, and I'll explain this in a moment when I get to what's called a seesaw model, they clamp the membrane potential to about the Nernst potential for potassium, which is the most negative potential that you can get in a healthy tissue. Okay, if you imagine that there is a way to fix the membrane potential in certain locations, you could also imagine that you can change the direction of current flow because you can fix one part of it to a very negative value. This is called the seesaw model. I'll get to that in more detail in a moment. So how do we look at the effect of blocking the potassium channel on featherbud morphogenesis? So depicted on the top row is, is a chick here. We dissect the skin out. We orient it in a specific direction for the head and the tail. And then we transfer this skin explant to culture. And then we image it and we treat it with different potassium channel blockers. In this case, we're using 4-aminopyridine and tetraethylammonium. These are well-known non-specific potassium channel blockers. Non-specific mean they don't necessarily choose one potassium channel over another. We have started to do work with specific potassium channel blockers subsequently. But anyway, shown below, and we'll show you real data in a second, is a depiction of feather buds in the back, the dorsum of the chick at H&H 31 and H&H 33 or 35. And you can see that, in the control cases, the buds get longer when we're not treating them with anything. If we apply the potassium channel blockers, we start to see changes. If we're applying the potassium channel blockers early, we see that some of these buds merge with each other and form these medial lateral stripes. And I'll show you that in a second. If we treat a later stage feather buds with, again, same potassium channel blockers, they don't elongate. And actually, they become slightly misshapen. So this is a figure showing control and then, different levels of the blockers. And what you see indeed is that instead of forming the normal, so this is control at stage H&H 31, and then waiting 24 hours to look at what's going on. So this is at time 0 and then progressively later. And we see the potassium channel blockers cause these strange stripe-like patterns. So potassium channels blockers applied early on cause changes in the pattern of the feather buds. If we apply the same blockers to a later stage, HH33, what happens is that the buds don't elongate. So you can see in the control case, this is HH33 times 0 after 6 hours and 94 hours. The feather buds elongate in the control case, but with potassium channel blockers, they don't elongate. And furthermore, they start to get these weird bumps on the sides of the feather buds. Another effect of the potassium channel blockers is that they reduce the level of beta-catenin and sonic hedgehog RNA. And what you can see this here. So this is at H 31 plus 24 hours, and then at 40 hours. And then you can look at, well, in the control case, at 3 different magnifications, you can see these are representing, the so-called placoid region where the feather buds are forming. And both beta-catenin and sonic hedgehog rise in the appropriate ways at where the feather buds form. Okay, and what we see is that the application of the potassium channel blockers leads the messenger RNA to fade away. Okay, so that's 24 hours and 40 hours. And that's true for both the beta-catenin and the sonic hedgehog. And that suggests that one of the effects, if applied early, is that these potassium channel blockers can interfere with feather bud, the success or not success in formation of buds. Okay, so we wanted to know what would we see if we applied blockers to the potassium channels that are, we think, located primarily at the anterior of the feather bud, where the current is flowing out of the tissue. And, the hypothesis is we have a standing current, and channels are located, different types of channels are located in different parts. What happens to the current if we block just one segment of this loop? And obviously, we're taking advantage now of our 3D vibrating probe. This is actually the envelope drawn around this vibrating probe, placed near, in this case, this is a single feather bud, and it can be positioned, of course, anywhere over the feather bud. We are going to measure in the anterior and the posterior selectively. Depicted here on the left is the feather bud, and the arrows are superimposed on the photograph to show where the currents are flowing into and out of the feather bud. So at this particular stage, which is H and H35, you see that there is current flowing into the feather bud at the tip of the feather bud and current flowing out at the base. Okay, and what we want to know then, as I said, is what happens if we block with the potassium channel blocker? Well, it turns out not only do we block the outward current, but we actually cause the currents to flip direction. So we actually see a current flowing in at the base and out at the tip. And this is reversible. So if we wash out the potassium channel blocker, we get a restoration of the flow in the same directions as in the original control situation. And we can plot this out, blue being anterior again, green posterior. At the beginning, before any treatment, the blue is outward current. So remember, that's positive current. The green is flowing in, so that's negative current. Then we apply the TEA, and you notice that there's a flip in the direction of currents. And then when we wash out the TEA, we get a flip back to the original directions. Um, so if you block the potassium channel current at the base, it also affects the current at the tip. Okay, so uh, this is really a very curious observation, but it's actually really important and relevant because if you look, the currents. So this is now from dozens of skin explants where Madison Zitting, who did this work, um, measured the currents indeed in the front and the rear of a bud and plotted this out. And it looks very similar to what was recorded previously by Min Zhao's group, although in this case, there is kind of a dip in the current, outward current here in the middle of this period when the current is flowing outward. And it's paralleled by a kind of a dip also in current flowing in the posterior. Anyway, but here's what's curious. And I mentioned this earlier when we were looking at Min Zhao's data, the current flips direction. In this case, it's around stage 37 where it's flipping. And this is not under the influence of any blockers at all. This is a natural progression in the currents during development. Okay. And this experiment here yields some information, which I will talk about in a moment in the so-called Seesaw model about what is going on. But it really suggests that this loop here requires all segments to be functioning properly. Okay, so what is this Seesaw model? So we know, again, this is really going back to high school physics, that a current will flow downhill, down a voltage gradient from the positive voltage to the negative. So current in this case on this seesaw would flow from the upper right to the lower left, as I've drawn it here, okay? The most negative potential is set by potassium channels. We can't rule out chloride channels so what's critical, and I won't go into detail about this, but what's critical is that these two types of ions have a Nernst potential that is around, minus 90. That is the most negative among the ion channels present in healthy tissue, all right? So in setting this gradient, that means that is the most negative potential you can have within the tissue. So here, looking at this, the negative region we think is at the base where there are potassium channels, okay? And the tip is going to be slightly positive charge because current is flowing into that region, which will depolarize that region, or that is make the voltage less negative. So there is a voltage gradient within the tissue, which leads current to flow within this tissue. Okay. Now, how would a change in direction occur? Well, if we could change which of these regions is more negative, then we could change the direction. Okay. And we haven't worked out exactly which channels are responsible for this change in direction, but we think we have a hunch. But anyway, we're still working on that hypothesis today. So that's pretty much what I'm going to talk about that's new. And we have ongoing work. Probably one of the more exciting ideas that we've arrived at is that the regions where the most growth is occurring within an individual feather bud those regions are called localized growth zones. And using different methods, we can identify where cells are multiplying, where different processes are most active. And these are the so-called localized growth zones. It turns out that the localized growth zones are where the standing current is entering the tissue. And, in retrospect, it doesn't seem that surprising because the processes occurring at the so-called localized growth zones are the same processes that calcium triggers. And we'll come back to this in a slide in a moment. The other thing we're doing is we are mapping out the ion channel currents in early skin development. So we're looking at right now skin that has feather buds on it, which, or I should say, placoed formation, so we can see the evidence for where the feather buds will be. And we identified that there are indeed inhomogeneities in the currents that are those inhomogeneities really reflect the underlying placoed formation, the early feather bud formation, and they mirror the positioning of these feather buds. And what we're doing now is we're working backwards to earlier and earlier times when we can barely discern where the feather buds eventually will be. And we're looking to see if there are already current inhomogeneities that precede placoed formation even. So this is what I mentioned earlier, the localized growth zone experiment. And using BRDU and then antibodies directed against it, and anyway, a whole series of steps, it's possible to visualize where at given feather buds at different stages. So this is called the short bud and this is called long bud, but the very active growth zones are located pretty much where we saw calcium and entering into the feather buds, okay? And if we look here, okay, essentially we see this current flipping at a specific time. And it turns out that the growth zones migrate from the tip of the feather bud to the base of the feather bud, okay? The so-called bulb or follicle base. So the growth zones migrate from indeed the very tip ultimately to the base. And if you look at the staging here, so initially the calcium entry is at the tip, but we get calcium entry at the base at a later stage. That coincides very amazingly to the movement in the growth zones. The other thing that we're working on, as I said, is looking at the currents that flow at the buds and around them at early stages, when there's really not, properly speaking, there's not real feather bud yet. And indeed, we're excited because this is Hans's work, because we actually now can make buds start to appear in the apteric regions by treating with a calmodulin inhibitor. And you can see this so-called apteric zone this is in the normal situation. Really, there are no buds, but treating with a calmodulin inhibitor leads buds to appear even in the apteric zone. So this is another tool or another observation that we can study when we compare skin in the different tracts of the early embryo. And we can look both at, for example, the region where there are feather buds, the region where there are no feather buds, and now we have these new feather buds, de novo feather buds, that we're going to study and characterize as well. And we think calcium channels play a role, not surprisingly. So I'm going to summarize. I've actually lost track of time. I'm hoping that I'm just finishing. And okay, so there are ion channels that do play critical roles in morphogenesis of the feather buds. They're important not only in early pattern formation, but they also shape feather buds once the feather buds have started to appear. There is, playing a key role in this, is a standing electrical current, which facilitates long distance tissue scale signaling, which is fast and reproducible. It serves to inject calcium ions deep into the mesenchyme. calcium is not only a current carrier, but also a second messenger, which plays big roles in some of the processes that we know are important in featherbud morphogenesis. There are localized growth zones, which we believe are located where calcium enters in the standing current into the featherbud. And we are looking to see, and we seem to find already evidence for inhomogeneities in the currents over the chick skin early in development. And we think these may be playing key roles in the evolution of or development of featherbuds. Anyway, that's it. The lab was carried out by a range of people within the Chong lab. The person who did a lot of the early work was Ang Li and Zhonghua from my lab, who did the patch clamp experiments. Madison did almost everything related to the potassium channel. And we have ongoing work that is being carried out largely by Hans and Zhou.